Howdy! We're ready for Chapter 18, Fun Chem 2. The boss down here. I can put that. So, Fun Chem 2, proteins. Alright, this is our last big chapter. We've got some small stuff left after this, but we're really hitting the home run here. So, looking at this chapter, a whole bunch of learning objectives. Don't let it intimidate you. Alright, so we'll start with our cellular functions of proteins. They're used for all kinds of things in the body. I'm not really going to be focused on those. I'm going to focus on you being able to start with this picture of amino acids. Remember, these are alpha amino acids. So we've got our carboxylic acid. We've got a carbon separating it from the amine. So this is our alpha carbon. The amine group is not directly attached to the carbonyl, so it's not an amide. Alright, we've seen this, I've drawn this many times, and this is the basic picture you want. This is the picture that allows us to see this R. You want this picture in your head. So when I look at this picture, this is, I'm going to star this, this is what we want. Now we'll draw this several different ways. It's just an acid. With an NH2 and some R. We could even draw it with the R over here and the NH2 down here. Doesn't matter. Alright? Now, we typically focus on the R. Okay, that's what determines which amino acid it is. We can draw these in Fisher projection. When we do this, the R is usually on the bottom, and it allows us to, we'll talk about these later, but it allows us to define D or L based off of the NH2. Alright, so this one is on the left, it's L, and so we'll play with those, but our home run picture, one that gets us all the points in this chapter, is this one. Alright, and there's a slight problem though. When I look in the book, I don't usually see things drawn like this. Instead, what happens is we've got an acid and a base. So carboxylic acids, pretty acidic, and we talked about amines as being bases. So these can react with each other, and if they react, we get this proton transferred from the acid side to the base side. So now I've got 3 protons over here. And I'll have a deprotonated acid. So I'll have a positive charge on this side and negative charge on that side. Now the whole thing is neutral, but it has both a positive and a negative in the same compound. This has a very special name. This is a Zwitterion. Zwitterion. Alright? And so Zwitterions then have both a positive and negative, but they're going to have equal amounts of positive and negatives. Alright? And so I'll ask you about these Zwitterions, and it's really just comes down to they're going to be deprotonated. So when you see these drawn either in the Zwitterion, what we would find in our body, or if they decide to draw it as the amino acid based off of our original picture, it's fine. So we'll see both of those. Now, here's Fisher projection, and so if the amine is, notice these are Zwitterions, alright? If the amine group is on the left, it's L. If the amine group is on the right, it's D. That's simple. Now, here's what we want to memorize, that in nature, the proteins are L. Alright? This is different because when we did, so proteins are L, and carbohydrates our sugar were D in nature. And so we want to be careful. You need to go ahead and just memorize these three. All right, so memorize this. I'll ask questions about them, and those become nice and easy problems. All right. Okay, so our proteins we can classify based off of the amino group. So this amine group, if it's on the left, it's L. If it's on the right, it's D. And so in nature, we want to keep up with, then the proteins will be L and our sugars will be D. So I will give you a amino acid and ask you to classify it as either being hydrophobic or one of these hydrophilics. Now usually what I'll do is I'll have hydrophobic, polar neutral, positively charged, or negatively charged. Alright, so we need to figure out then, how do I tell those apart? Well, if it's hydrophobic, remember it doesn't like water. It's carbons and hydrogens. R has all carbons and hydrogens. Nothing else, no charges, alright? And then if it's positive and negative, you just add up the charges. So if it has a Zwitterion, remember the positive, so our H3N, here's our picture. This positive and that negative cancel out. But if our R group has some kind of positive or a negative on it, then, for instance, if it had a negative charge here, we would be able to say this is a negatively charged amino acid, and so it's going to be water soluble. If it's positively charged, it'll be water soluble as well. Then our last category then is this polar neutral. And what are we looking for there? Well, R has S's and N's. Alright, and we won't see any, but if it had a P in it, phosphorus, anything besides carbon and hydrogen. So we're looking for oxygens in there, anything that's not just carbons and hydrogens. Alright? Alright. So let's look at a list of these. Here's a picture out of the book, and they like to give these in categories. So as I look at these, I see all these amino acids. It's our basic picture. We're just really looking at the R. And I'll give you a easier, I don't know about easier to read, but a different picture. And I don't really like this way they've classified everything here. It's kind of confusing. So I'm going to scroll down here and look at picture I would use. And so this one, I'm just telling you R. And so these are only really useful if we draw an amino acid out. So I'm going to draw one out, and while I'm doing that, so if this was, for instance, this compound, we could come in and we would look here. There's our R. It's a CH2SH, and so that's going to be cysteine. And you'll notice that each of these has, so that was cysteine, each of these has a 3-letter symbol, and it's got a 1-letter symbol. Alright, and so we're going to usually focus on the 3-letter symbol for us. Alright, and so I've got these listed out, and you will have this on the test. So you'll want to print these out or have them available for you to look at during the test. That's fine. Alright. The only ones that are really kind of difficult here is proline. You'll notice here's my alpha amine. my alpha carbon, and it wraps around and makes a ring. All the rest of these, it's just hanging off. So our simplest case then is, wait a minute, this one's hidden in here. Yeah, our simplest case then is just when R is just a nice methyl group. All right. Oh, sorry, right here, glycine. If R is just a hydrogen, so that's our only one where that alpha carbon. It's the only one where this alpha carbon is not chiral. All the others, that's a chiral carbon, and so that's why we can do our DNL assignment. So on these, if I want to do the D and the L, I'll give it to you in Fisher projections. So that makes it pretty straightforward. All right, let's go back to our ugly pictures here. So here's all these pictures. You don't have to memorize these. You need to be able to look at them and classify it in terms of so when I go back and I'm see one of these drawn as a Zwitterion or not, and I look at this one, and so here's my Zwitterion. I'll ask you the name of this one. I'll even ask you then to classify this one. All right, so this one, it's got an OH. That means it's going to be, it's not hydrophilic. It's not hydrophobic. So, and it doesn't have a charge. So this will be neutral polar. All right, and that's because of this OH. So any kind of thing other than carbon or hydrogen, it's going to be neutral polar. If it's got a positive or a negative, even if it has one of those oxygens or sulfur, you know, then it's going to have, you'll tell me which charge. So I'm not worried about this acidic versus basic. I'm going to be going by our categories. Is it hydrophobic, or is it polar neutral, negatively charged, or positively charged? All right, so you'll be able to look at the picture and tell. All right, so amino acids themselves are okay, but when we start linking them is where we really get to forming proteins. And the linkage here, it's a peptide bond, but it's really just an amide. So we're taking one amino acid and reacting it with another one, and so it's the amine group that reacts with the carbonyl. And the key to this amide, is this repeating pattern. So looking at my peptide bond, this repeating pattern as I form these. If I take two of these, I'll have NCC, and we can see that the next group, this N, will react with this carbonyl. So when we're done, I'll have this NCC, NCC. And so that is going to be our key structure, is NCC. So if I get a whole big chain of these, we can figure out which ones are which based off of where this R is. Remember, the R is always off of that middle carbon, and it's going to have this repeating pattern NCC. So keep saying that one to yourself. Now, we call once we get a whole bunch of these, you're going to have one end that has the amino group and one end that will have the carboxylate. All right, so our carbonyl end is the C terminus. It's just ends in the C. And so, but I'm looking for this repeating pattern, NCC, NCC. So if this N was 1, then I could just go 1, 2, 3, 4, 5, and count the N's. And that would tell me this has 5 amino acids in this protein. All right. Now the order, you always name it from the left to the right. So you always start from the N terminus to the C terminus, even if they flipped it around and wrote it backwards. So if we were going to name these, you would look up the R, and so everything's considered a derivative of the full name, but we don't really. So you drop the YL, drop it and add YL, and you do all that stuff. I'm not going to hit you with that. What I'm going to be hitting you with is asking for the order. And so you can give their full name, or you can give, when we look at these pictures of these, then remember each one has these three-letter uh, symbols. And so you can use that to tell me what protein it is. All right, so here's one, and we'll ask a couple of different things about it. So I'll start off with this: how many protein or how many amino acids are here? So let's count them. 1, 2, 3, 4 on the first one. All right, and then as I'm going through these, I might ask what their order is, and so we would come back over here, and we would do a matching basically. And so uh, so I'd find CH2. So I'm gonna make a list. So I had CH2SH, that's my first one. The next one was a CH2, CH2CO with a negative. This one is, so this one was polar neutral. This one will be negatively charged. Then I had a CH2 and then a benzene ring. All right, and then the next one, remember I'm looking NCC, and I'm looking for what's on this C. I'll put an H up here, but we don't care about that H. All right, so this one had an OH here and a CH3 there. All right, so now I'm going to go to my list, and I'm going to use the one that you'll have on the test. So that's way down here in the bottom of my PowerPoint. Hang on. All right, so CH2SH, here's CH2SH, so that's CYS, so cysteine, and then I had CH2, CH2O negative, there's the CH2, CH2, and then my carbonyl, and it's deprotonase, it's GLU, all right, and then I'd had CH2 with a benzene ring, don't see that on this screen, so the next picture, oh, here it is. Now this little zigzag, notice the little zigzags here, that's my parent backbone. So it's just showing where it's attached. Alright, so this is phenylalanine, I'm sorry. And then the next one had like an isopropyl, there it is, and that's 309. Oh, it likes that, 309. Sounds like I'm gonna call Jenny or something. Alright, so that is THR. So we can classify these. It would be cis, glutamine, phenylalanine, 309. When you're doing these, keep in mind, go back up to our practice. When we're doing these, so when I was doing these, I always start with the N side and I'll work my way to the C side. All right, and so the N terminus, I might just say, what is the N terminus? And so it was cysteine. I'm going to ask what the R terminus was. Or I may say, what is the order? And so it was cis, glu, phenylalanine, 309. Okay. Or I might circle one and ask. So this one would be, that would be neutral polar. This one would be hydrophobic. This would be negatively charged. This would be neutral polar. Looking on this one, I've got this one is neutral polar, hydrophobic, hydrophobic, neutral polar, hydrophobic. See that H? It's a sneaky one. And then hydrophobic. All right, so this one, let's count. 1, 2, 3, and let's get a little bigger. 1, 2, 3, 4, 5, 6. 6. Yep. All right. So first thing I'll do is I'll look at those pictures, and I'm going to over here, and I'm going to look at this picture and I'll draw what the R is. So squiggly line, I got a nice little purple group. Then I got R is just H, and then I got a benzene ring. It's a phenol. And then I have R is CH3. And then we got benzene, just regular benzene ring. It's got that CH2 though. And then I've got a CH2, CH2S, CH3. And so I'll go through my list down here. And I'll look over on this and start with this CH3. And so I'm looking, hey, that's this one way down here, kind of hidden. All right, and so that's Val. And so I will come over here and write, the first one was Val. The next one is that R is equal to H. And so I find it on the list. R is equal to H. That's this one. Glycine is always. All right. Next one we had benzene ring. OH. That's here. Then we had fourth one. R is just a CH3. Alanine. Then we have PHE. And then the last one down here had that sulfur. That's pretty distinctive. That's MET. And that would be the order. Now, 1, 2, 3, 4, 5, 6, just like we said. All right. So on these, you don't have to try. I'm not going to have you try and figure out how to change the spelling of these. We're going to be, what order is it? Asking you details about these, and then I'm going to ask you to list them in order. Or I'll even ask, what's the N terminus? What's the C terminus? That kind of stuff. So everything, remember, was based off of that picture. All right. So now we're ready to start talking about the structures. And so the structures of proteins, we've got 4 structures that we're going to worry about. All right. So we'll start with this picture. And so this is going to list our 4 structures: primary, secondary, tertiary, quaternary. All right, let's tell how to tell these apart. So this is a great picture over here. This picture really kind of puts in order. All right, so primary is going to be just the order of the that we were just doing, the order of the amino acids. And so that's the primary structure of our protein. And then secondary will be the how it twists, how it folds. And so it'll either be a helix or it'll be a pleated sheet. We could get random coils, but these are our, we'll spend some time on those. Excuse me. And tertiary is going to be these advanced folding. And so it's going to be, that one's a little bit tougher, but it's going to be not just how it twists, but it's going to be these large scale folding. So I'll demonstrate that one. And then the quaternary, usually it's going to be more than one protein and they come together. And here's the key: quaternary includes the function. What is it doing? What is its job? All right, so get handy thing here. All right, so we're going to start off. We just got a rubber band. Okay, and I'm going to start twisting it. So the order of the chain on my rubber band would have been the primary structure. This twist is a secondary structure. So this one's twisting, no problem. Now if I keep twisting it, I'll get these large bends. And the easiest way for me to demonstrate is just actually to come in and you can see those large bends. That is the tertiary structure. Alright, so when you keep twisting this, you get those large bends. That's the tertiary structure. And if I took a couple of rubber bands and mixed them in together and they were all twisted in together, that would be our quaternary structure. But when I ask these questions, the quaternary structure will always include the use of that protein. All right, its job. All right, so primary structure, you're just going to list them out. So that's easy enough. That's what we were doing with before. Secondary structure then is how it folds, and so alpha helix has this pretty looking picture, and it's all held together by hydrogen bonds. They're intramolecular hydrogen bonds, right? So now if it comes in and it doesn't make that twist, it tends to make sheet. So this is a beta pleated sheet, and it's got these folds. It goes up and down, but it's again held together by hydrogen bonds. All right, so pleated sheets and helixes, those are pretty easy to tell apart. All right, so we can, when they come in together, they can either be parallel or anti-parallel. So parallel, then the N-terminus will be completely lined up. So if you think of the N as kind of being the head of the, it's the positive charge, it's the head of the protein. Then if their N-terminus is lined up with the C-terminus, then it's going to be anti-parallel. All right. So that's easy enough. I don't ask too many questions on that. Tertiary structure, we said it was those big large bends, and this is where a lot of biochemists spend their time studying, is mapping out these tertiary structures, because this is what determines where all the acids are going to be, so that a molecule can come in here and interact with them, and it really comes down to where these bends are as to what fits in there. All right, so quaternary structure, we might have more than one protein, and it's going to involve its use, its job. All right, now what causes problems with proteins? We can denature them. So we talked in lab about denaturing alcohol, and we said we poisoned it. All right. Well, if we denature a protein, it breaks apart all those hydrogen bonds, and it causes it to become unorganized. And so high temperature, pH, organic solvent. One of the big ones though is detergents, and so that one can really break down a bunch of proteins. Heavy metals, so it can really cause havoc. And then mechanical stress, if you just were to grab one end and pull it apart, you would, if you applied enough force, you'd break those hydrogen bonds. So denaturing is usually really, really bad. So you can, when you wash your hands, it's big going on now, you might have heard that you really should wash your hands for at least 20 seconds. Well, you should also wash them kind of twice. So you wash them, scrub them real good, and you're denaturing all the proteins and killing off all the things that are in there. You're destroying the proteins. And then you wash them again really good, and that's the detergents doing their job. And then you wash them again and wash away all the extra remaining stuff. All right? Anything that after it's broken apart you want to get rid of. All right, so dietary proteins, so when we eat these, they come back down into amino acids, and we can synthesize all but 9 amino acids. And so when they are called essential, that means we can't make them. So you have to be careful, because the essential amino acids are things that we have to have that we can't make. Our body cannot make these. Now, you may have also heard of essential oils, and essential oils are just the things that give the smell or the flavor of whatever they're taken from, usually a plant. So essential oils of cloves, all right, and it's just the thing that gives it its smell. And so it's not that it's necessary for your body. So be careful. Essential amino acids, we need that. We can only get through our diet. Essential oils, not so much. So they use that language differently. All right. So a complete protein has all the essential acids that we need. So meat does that. So you got it. So a lot of times people say, hey, if you just eat vegetables, you get stuff, but you need to take a protein supplement. All right. So you got to have those necessary proteins. All right. Howdy. Welcome back. So had to take a little break there for biochemistry. My dog had a biochemistry of its own. So DNA, RNA, we'll be looking at these, but really what we're focused in on. So we've got each one of these has the sugar, which either, depending if it's DNA, then looking at the parts here. So here's our ribose, and if we take this oxygen off, this OH group here, it's deoxyribose. So DNA uses this sugar, RNA uses just ribose by itself, and we're taking a phosphate group and attaching it on here to our CH2OH that we always have sticking up, and then a base goes on here, so one of the nitrogens attaches and replaces this OH. So the key to this is each base is coded, and so it codes over to one of the each base codes to one of the amino acids. So here's so again, here's ribose. It's missing this OH down here. Right, it's this bottom right. So this would be for DNA, and so here's our phosphate linkage, and the next sugar over will be one of these, and then we've got so here's our base, and so I'll leave you to deal with all of the biochemistry. You're capable of reading the chapter. So what will I be testing you on? And so A to T, C to G, I'll let you read up on all of that. But what I really am worried on is how to read one of these codons. And so the codon codes is what we're after. And so I'll give you a series of these, and it'll give you the base. So for instance, I might pop up something like C, C, A, C, A, C, G, A. And so the key to this is it's 3. So you find 3. The first one, base, you'll find on your list. And so if I pull up the whole list, you'd be able to go through and so here's what this list would look like. And so I'd find the C on the left. I'd find the second one is in the middle here, so you're going to follow it down. And then once I've done that, that kind of narrows it down. So this whole section is C's. These 1, 2, 3, 4 are my seconds. So these are my possibilities. This column here, and then I can narrow it down. So here's my C. So we're looking for CCA. So here's the A, and then for E's, I've put all 3 of them here. So this is, first one is proline, and then I can find CAA. So we'll find the C. I'll come over to the A's. CAA is glutamine. and then CGA. So we find the C, come over to the G, GA, and arginine. And so this literally just tells us the order of the proteins that we would be transcribing. And so some of these are as simple as telling us when to start and when to stop. And when it actually hits the stop, it lifts off. So this is just going to be really simple. I'll give you a series of three, and you just follow this easy little code here. So you'll have this handout, you'll print it out and use it. All right. So it's just on the one I give you, it even has the 3 letters here. So the book has one, but it's not near as handy. All right. So the end of this chapter is not real bad, and so we've just got quite a bit of memorization in terms of the protein, the 4 primary, secondary, tertiary, quaternary, and then you've got to learn how to do the Zwitterions. You got to also memorize proteins are going to be L, carbohydrates for D, and then you've got to be able to apply your knowledge and then of looking at the R group and deciding what does the R look like. So a lot of stuff that we're just going to be doing skill based, and so it's based off of a couple of those handouts. Then we'll have this handout as well as the handout that lists all the amino acids. All right, so we'll get homework on this, and then we've got, I think, one more chapter to go. Howdy. So last chapter here, chapter 19, enzymes. Really, all I really want you knowing about enzymes, they're catalysts, all right, and they speed up a reaction. So we've talked about catalysts, and they do that by lowering the energy of activation, all right. So that's our main thing that we'll be dealing with for these catalysts. So that's what I want you to know. And enzymes do. They deal with lock and key, and so they only deal for things that fit into that enzymes and the binding site determined by the tertiary and quaternary things of the proteins, or what allows these enzymes to get in there and fit in. So only certain substrates will fit in there. Alright, so then we also need to know our definition for vitamins. And vitamins are things that you've got to get through your food. Alright, and so and one of their main uses is for coenzymes, and so that helps fit in here and put the right substance into our enzymes. Alright, so that's the main thing. We're just going to enzymes are going to speed up the reaction.